Hi, my name is Jamie Dean, and I just joined the City of London RadNet Centre as a junior group leader. And my team are based at UCL in the Department of Medical Physics and Biomedical Engineering and at the Cancer Institute. So my lab is working on computational radiation biology and oncology, and we're interested in three main areas. So firstly, how cells are resistant to radiation. So whilst radiation is very effective at killing some cancer cells, others are able to survive the treatment. And so we want to understand what is it with those cells that make them resistant to the treatment. And then if we can work that out, can we design new treatment strategies in order to target those cells? The second area we're interested in is how to optimally combine new targeted drugs and immunotherapies with radiation. So there's a lot of interest in these new combination therapies, but something that hasn't been worked out very well is how you should administer these treatments in combination. So what doses of each you should give, what timings you should give them at. So we're interested in working out that. And then a third area is trying to better personalize radiotherapy to individual patients. So both in terms of coming up with personalized strategies that increase the efficacy of radiotherapy and also reduce the side effects from treatment as well. So taking each of those areas in turn. So for radiation resistance, we're particularly interested in non-genetic causes of resistance. So there's a lot of interesting work going on looking at how genetic mutations that occur can lead to resistance to many different cancer therapies. But even if you have a population of genetically identical cells and you treat them all equally, we know that some of those cells are able to survive the treatment. And so we're trying to understand exactly what aspect of those cells is making them resistant. And so we're particularly interested in noise or randomness that goes on in cells that can cause some cells to have slightly higher levels of certain proteins, or other cells to have slightly lower levels of certain proteins. And then we're particularly interested in the dynamics of key proteins that control the transcription of genes that are involved in cell fate decisions. And so we're interested in how the dynamics of these proteins differ between different cells and how that can lead to different outcomes. And then also trying to understand how the dynamics of these proteins can be manipulated either through novel radiotherapy treatment schedules or through novel pharmacological strategies in order to alter the dynamics of these key proteins in a way in which it will steer the fates of these cells towards desired outcomes, such as cell death. So in some cases, the important proteins haven't been fully worked out yet. And so in an initial stage of the work, we take an unbiased approach to try and discover what the important proteins governing cell fate decisions are. In other cases, these proteins are much better understood. So a classic example is the tumor suppressor gene TP53. So it's known that TP53 protein undergoes a series of oscillations following double strand breaks, which are induced by ionizing radiation. And it's been shown that if you can manipulate the dynamics of the TP53 protein, you can alter the cell fates. And so we hope to uncover similar kinds of key protein dynamics and ways to manipulate them that can similarly lead to being able to steer the cell fates in desired directions. In this kind of more fundamental radiation biology research, it's a bit challenging to know exactly how this will change or potentially change clinical practice further down the line. But the kind of things that it could lead to would be new ways of dosing the radiation that can impact on the dynamics of important proteins. And then another thing is that if we can understand the networks that these proteins are involved in, we can understand how to use new drugs in order to manipulate the dynamics of those key proteins. And so it could lead to new drug radiation combination therapies that would then be tested in the lab first. So one that we're working on a lot is glioblastoma, which is the most common and aggressive brain tumor in adults. And unfortunately, survival from these tumors is extremely low. They're very resistant to both radiotherapy and chemotherapy. And so there's really a dire need to come up with new treatment approaches for these tumors. So that's something that we're working on a lot, and collaborating with experts in glioblastoma biology on to try and see if we can understand the biology of the dynamic response of these tumors to treatment, and how we can potentially come up with new treatment strategies that take advantage of this improved understanding of the biology. Yeah, so our second area, which is focused on trying to optimize treatment administration schedules for these novel drug radiation combinations. When you combine a drug that's designed to improve the effectiveness of the radiation, so those are often drugs involved in inhibiting some aspect of the DNA damage response of the cancer cells. It's very hard to know exactly what time you should give these different treatments at and what doses you should give. So currently what's done is that the drug is given in the same way as it would be given either by itself or in combination with chemotherapy, and then the radiotherapy is given in the same way as it would be given by itself. But this is highly unlikely to be the best way of administering these treatments. So when you have these combination therapies, there's an infinite number of possible ways you could administer them. And in order to try and come up with a better administration schedule, what we do is develop mathematical models of a tumor response to these different treatments, using experimental data to inform our models. And once we have a mathematical model that accurately describes the response of these tumors to these combination therapies, we can then simulate in the computer many different treatment strategies. So essentially we can perform virtual clinical trials comparing different administration schedules, and then once we've found the one which is predicted to be the best by the mathematical model, we then work with experimental collaborators to test those new administration schedules first in the lab before adapting them for clinical use, and then testing them in early phase clinical trials. Timing is particularly important in the context of drug radiotherapy combinations, because the timing of the DNA damage induction following a radiation treatment is very different from a drug treatment. So with a drug treatment, you'll get a gradual accumulation of the drug in the tumor, and then a gradual washing out of the drug over time. Whereas with radiation, we administer all of the damage in a much shorter period of time. So then it becomes much more important to get the timing of the drug and radiation administration correct in order to maximize the interaction between the two treatments. So the third area, which is focused on personalizing treatment to individual patients, there are a few different aspects of this that we're interested in. So firstly, in terms of enhancing the efficacy of treatment, we want to use a combination of mathematical and statistical modeling approaches combined with longitudinal monitoring of patients' response to therapy using either imaging or biological data in order to make a set of initial predictions for how an individual patient will respond to a set course of treatment. So then we have a prediction for an individual patient that changes through time over the course of treatment, and then the other thing that we want to be able to do is use these data to develop mathematical models of treatment response again, and that will allow us to predict the impact of changes to therapy. So that could be either selecting a different treatment, or it could be adjusting the administration schedule of a treatment. And so then over the course of treatment, we'll get not only updates of the predicted response of a patient to the standard treatment, but also some predictions for what the best treatment approach for a particular patient at a particular time might be. So this kind of approach still needs a fair bit of work, but we hope this is something that we can make a lot of progress on in the coming years. And then the other area we're very interested in is reducing side effects from treatment. And so I'm interested in understanding how different regions of normal tissues that get irradiated during the course of treatment have differences in their sensitivity to radiation. And then also how that sensitivity to radiation can vary, not only as a function of the spatial location, but also with age as well. So in particular in the treatment of pediatric patients with radiotherapy, because these tissues are still developing over time, their sensitivities to radiation are also potentially changing over time. We're investigating using some machine learning approaches to try and predict how different normal tissues vary in their sensitivity to radiation throughout developmental time. And so what this will allow us to do is, once we've created these maps of the spatiotemporal radiation sensitivity, is that it will allow us to recommend new radiotherapy treatment planning approaches that are designed to minimize the dose administered to these most sensitive areas. And it will also guide which patients might benefit from newer radiation administration technologies such as proton therapy in order to reduce the dose to these sensitive normal tissues and hopefully reduce side effects from treatments. For me personally, the most exciting aspect of radiotherapy research at the moment is really the coming together of many different fields that I think is really important to make progress and lead to better treatments for patients. So bringing in the latest expertise and technologies from molecular biology, from cancer evolution, from physics and engineering, and systems biology. And I think now we're starting to see these new areas which are coming from bringing in these ideas from other fields. I think is a very exciting time, both the fundamental biology of the radiation response, but also in how to improve our translational research and clinical research as well. So for me, that's the most exciting thing, working in this highly multidisciplinary environment, which can lead to hopefully lots of new and helpful insights that none of these fields would be able to make in isolation.